Thank you. First, we would like to apologize for the problem with the schedules because we skipped someone in the morning, so I have to postpone my presentation later. I'm not going to talk much, about 10 minutes, just to go and schedule. So I'm going to talk about siRNA and how can we use it to control whitefly. We know that plants can be infected with a lot of pathogens like bacteria, virus, and so on. And the most devastating is the virus because we cannot control it by using pesticide or so on. Once it's inside the plant, it's very hard to control. So if we can find a way to control this virus, that will increase the yield, and we can reduce the use of pesticides that we use it to control the vector that transmit the virus. I'm going to talk about Geminivirus. As we know, Geminivirus is a DNA virus, a single-stranded DNA. The one we are concerned about, it's transmitted by Whitefly Mysia tabasi, and it infects a lot of crops, especially dicots. And this virus is highly spread all over the world. And the number of viruses increased since 1977 until now. It tripled the number of the virus. It caused a lot of losses. It can reach $5 billion per year in tomatoes and $1 to $2 million in cotton. In Egypt, we don't have a problem for Geminivirus. Our cotton barbadensis is resistant to this kind of virus, but it caused a devastating problem for other countries that use hirsuta. So the TYLCV, which infect tomato, is tomato yellow leaf curl virus. It first started in 1939 in Palestine. Then it was found in India, Sudan, and finally in Egypt in 1969. This virus has one per type, one circular DNA, single-stranded. Once it enters the cell, it forms double-stranded. It has 6 operating frames. We have early genes and late genes. Early genes and late genes. The early genes is the Rib genes, the only gene that is responsible for virus replication produced by virus, but the virus use the replication apparatus of the cells to replicate. And we have C2, C3, and C4. C4 is very important because it's responsible for movement of the virus from one cell to another. This is how the virus looks like under electron microscope. This is the whitefly that transmits the virus, and it causes severe damage to the plants. So Geminivirus is very difficult to control, although they produce by breeding strains that can resist a little bit the virus, but after 1 or 2 years the virus break the resistance and start to outbreak again and cause problem to the plants. This virus can infect also Solanacea, not only tomato, and there is no R gene found that can control this virus or resistant gene produced by plants. So the only way to control this virus is using genetic engineering. Here we can see the whitefly can be found with a lot of population, and this is not a cloud. This is a whitefly that infects cotton in the United States. So it's a huge number of insects. And in Egypt, we have one of the most important crops is tomato, because we have a lot of production and a lot of area is cultivated with tomato. So tomato yellow leaf curl virus is the main problem in cultivating tomato in Egypt and all the Mediterranean region. We used before several strategies like co-protein-mediated resistance, antisense for Rib gene and movements, and even we expressed the C4, the movement protein inside the plant. All of them, the virus was able to break this kind of resistance. So we used the RNA interference. The RNA interference, when we use it before, we use, maybe we have some picture. Okay. This is the genome. So we use the different region of the virus. We use the C4 is overlapped with the Rib gene, and we use this region between C2 and C3, and between C2 and C2 and Rib gene and C2 and C3, and the pre-coat and the core protein. We tried each one alone using a fragment about 70 to 120 base pair. The best strategy was in C4, C2, and C3. So I wanted to use this, but we have a problem that the virus itself can break the resistance for the post-transcription. The siRNA strategy can break the dicer. So we switched to another strategy that we use small hairpin structure. We only synthesize a very short fragments of the siRNA with the region, an intergenic region about 9 nucleotides to form small hairpin RNA. This way, the dicer rule is very limited, and then we use it for resistance for the 3 structure that we have. Here, the Whitefly, when it fed on the fly and the liquid of the leaf, it injects with it the virus, and the virus can move from one cell to another. So, as we said, we choose 3 parts. The one in the beginning of the C4, the movement protein, and the region between C2 and the rib gene, and between C3 and C2. We have constructed we use different programs to design this RNA strategy to choose the best one for synthesizing this structure. And we already have synthesized 2 primer. We don't have to clone the gene. We synthesized about 65 nucleotides, single-stranded, and put them together with the sticky end of the restriction enzyme and just clone it under the control of type 3 promoter. And in this case, we use 7SL promoter from Arabidopsis. So once it's transcribed, it forms double-stranded and form small hairpin, which form at the end siRNA. We use in Agrobacterium the 3 construct together, so we put the 3 region in one construct between the left and right border without 35S promoter or terminator. In the beginning, we tried to use agroinfiltration to see if this strategy worked well or not. So when we try agroinfiltration with the syringe, it was very hard to use it with tomato and other crops. So we prefer to use a vacuum. As we said, we use a 7SL promoter to produce our siRNA for the 3 genes, to control 4 genes, really, because we have the rib gene, C2, C3, and C4 gene. So we use 3 constructs to control 3 genes. And after agroinfiltration, we started to detect for the viral genome after infection. The double-stranded RNA start to appear after 3 days, but after 2 weeks, it was very clear. So the region we choose, some of them was in region of the gene and other in the C region of the gene. This is the RNA, and we can, after 3 days, we can hardly see the hybridization, but after 2 weeks, it was very clear, and there was a correlation between the intensity of Norton hybridization with the virus presence in the cells. Now we know that the strategy worked well, so we switch it to stable transformation. We use Agrobacterium mediated transformation, and we produce plants with this strategy in tomato. When we choose this part of the genome, we try to choose a common region, a conservative sequence in all Whitefly transmitted Geminivirus. So we will not only control the TYLCV, we can control other type of Whitefly transmitted Geminivirus. And fortunately, we have another virus in our lab. We isolated. It's strawberry leaf curl virus. It has high similarity with the TYLCV. And we started doing some tests for this virus. This is the strawberry leaf curl virus. As we can see, this is the infected plant, and this is plant was propagated from virus-free, so it has some type of resistance. That doesn't show the virus because they cultivated after as a leaflet, so it skipped the infection. And here the strawberry leaf curl virus, it had the same sequence, but it has a shortage or deletion in the C3 sequence. This virus infects also tomato, but it has very mild sequence, but it causes damage for strawberry. This is healthy plants. So first of all, we started to do establishment for regeneration and transformation in strawberry, and we also did agroinfiltration with the same constructs, and we see that this virus was also effective in strawberry. Right now we are in the process of completing the transformation with strawberry to get strawberry resistant virus. And thank you.